All right, let's jump right into one of the biggest puzzles in all of biology. How does a protein get its shape? We're going to tackle this by looking at two totally different ways of thinking about it. Think of it as a competition. One method is like an architect, building everything from the ground up. And the other is like a detective, sifting through evidence to find a match. So it all boils down to this really fundamental question. You see, a protein, when it's first made, is it's just a string, a long, floppy chain of little building blocks called amino acids, kind of like a beaded necklace. And honestly, in this form, it's completely useless. But then, something amazing happens. That string twists and turns and folds itself into a mind-bogglingly complex, very specific 3D shape. And that shape? Well, that shape is everything. It's what turns that useless string into a tiny, life-saving machine that can do anything from fighting off a virus to carrying oxygen through your body. But get that fold just a little bit wrong, and the whole machine just breaks. So how in the world do we predict that final, perfect shape? Well, let's meet our first contender, the architect. The scientific name for this is Ab-initio modeling. It's Latin, and it literally means from the beginning. And that's exactly the philosophy here, to build the entire structure from scratch with no cheating. This whole idea rests on a famous discovery by Christian and Finson. The core belief is this: that simple string of amino acids, it already has all the instructions it needs to fold correctly. The blueprint is baked right into the code itself. It doesn't need any outside help. It just knows what to do. So the architect's method is, super pure. It takes that amino acid sequence and just applies the fundamental laws of physics. We're talking things like magnetism, energy, atomic forces, to calculate what the most stable, lowest energy shape would be. It doesn't peek at other proteins to see how they did it. It solves the puzzle from first principles, every single time. Okay, so what does that look like in practice? It's basically a three-step process. First, you have to describe the protein using math. Step 2, you use these things called energy functions to figure out how stable any possible shape would be. And then, the big one, Step 3, you unleash an army of computers to start an exhaustive search for the one, single shape that has the absolute lowest energy. And let me tell you, that search is the real beast here. The number of ways a protein could fold is astronomical. You have to imagine this massive, rugged mountain range with millions of valleys. The protein's correct shape is the single deepest point in that entire landscape. Finding it is a monumental task. To even attempt it, scientists use these incredibly powerful computational strategies to explore that crazy terrain. All right, so that's the architect. Now let's meet our second character, the detective. This approach, known as threading, couldn't be more different. Instead of building something new, the detective is all about finding patterns, sifting through old case files to see if this new mystery matches a crime that's already been solved. The detective's whole philosophy is based on this one super powerful idea. Nature is kind of lazy, in a really smart way. It doesn't invent a totally new protein shape every single time. Why would it? It reuses successful designs over and over again. So the bet here is that this new, unknown protein probably folds into a shape that we've actually already seen before. Now, every good detective needs a rogues gallery, right? A book of usual suspects. For protein threading, that's a huge database of all the known protein folds. That library has, get this, just under 1,400 unique shapes that act as clues. The detective's job is to see if our new mystery sequence can fit or thread onto one of these known templates. And the detective's method? Well, it's also 3 steps, but it's a completely different game. First, you build that library of known folds. Second, you have to design a scoring system that can tell you how good the fit is between a sequence and a shape. And finally, you basically try on every single outfit. You thread your new sequence through every single one of those 1,400 folds to find the one with the best score, the perfect match. Okay, so we've got our 2 players on the board, the architect, building from pure physics, and the detective, matching against existing evidence. Let's put them head to head, architect versus detective. Let's see how they really compare. So the biggest difference really comes down to where they get their information. The architect is a purist. It only uses the sequence and the laws of physics. It's completely independent. The detective, on the other hand, is a pragmatist. It's totally reliant on that library of known folds. And that's its Achilles heel, right? If a protein has a brand new shape that evolution just cooked up, the detective is completely blind to it. It has no clues to work with. This all leads to some really big trade-offs.
The architect's approach is incredible because it can, in theory, solve any structure and even show you how it folds. But man, is it slow. We're talking supercomputer level slow, and it really only works for pretty small proteins. The detective is way faster and has a pretty good track record, but its big weakness is that it assumes we've already seen all the possible folds, and sometimes the match it finds is, well, it's a bit good enough rather than perfect.
So, who wins, the architect or the detective? Well, here's the really cool part. It's not about one winning. The real future, the absolute cutting edge of this field, is when they work together. It's about combining their strengths. Take a look at a program like iTasser, which is one of the top prediction tools in the world. It's a hybrid, and it's brilliant. First, the detective comes in and does some quick police work, using threading to find a few solid leads, some possible shapes from the library. This gives it a fantastic starting point. Then, it hands the case file over to the architect. And the architect gets out the physics books to refine that initial sketch, perfecting every little angle and bond. The detective finds the lead, and the architect cracks the case. It's the best of both worlds. And that really brings us to the big picture, doesn't it? As these incredible architect-detective teams get faster and more accurate, we get closer and closer to being able to predict the shape of any protein just from its code. And when we can do that, what happens next? I mean, think about it. We're talking about designing brand new drugs from scratch, creating custom enzymes to clean up our planet. We're talking about unlocking the secrets to the machinery of life itself. The puzzle is still huge, but wow, the tools we're building to solve it are just getting better every single day.